So now that you know a little bit more about what a genome is, what it means to sequence a genome, and why that's an important problem, I wanted to come back to this newspaper problem that I had mentioned before. So if you were given enough time to figure out what all these little pieces of paper said, and somehow put them back together into knowing what the news was on that day, how is it that you might solve it? That's the question that we want to think about. So ignoring that we don't see a connection to biology at this point, but just thinking about this brain teaser, we're going to think about how might we put these pieces together. And there's a critical insight here, which is that the newspaper problem is not a jigsaw puzzle. So that's for a couple of reasons. For one, well, we've got multiple copies of the same edition of the newspaper. With a jigsaw puzzle, you have one picture that you're putting back together. With the newspaper problem, you have multiple copies of that newspaper, and you just need to know one of those newspapers. And for that matter, you're never going to be able to maybe put one copy back together because some of those pieces probably were lost. So maybe some of them burned up or were blown to bits in the explosion. So there's not one newspaper out there where all the fragments from that newspaper are still there. So instead what we have to do is that we have to use overlapping shreds of newspaper in order to figure out what the news was. So we might imagine that if you had two pieces that overlapped in 20% or something, then you can start to overlap those and glue them together, and then you saw another piece that overlapped with this one. And so this gives us a giant overlap puzzle. In fact, you could probably, in the case of the newspapers, you could use other context clues. So you could say, well, I know that this is a sports article, so it should go in the sports section and things like that. But even if you just use the overlap, you could still solve this problem. So imagine it were a newspaper that were written in a language that was foreign to you. Okay, you still could solve the problem based off the overlap. So the question is, what makes genome sequencing so hard, and how does it connect to this? Well, when you read a book, you can just read the book one word at a time from beginning to end. But modern sequencing machines have been able to figure out what the sequence of nucleotides is on a piece of DNA. But the critical point is that they can only read, and they're able to read this without actually seeing the nucleotides, but they're still able to indirectly infer what it is that these nucleotides are, but only if the fragment of DNA is very short. So these short fragments of DNA that the sequencing machine can read are called reads, fittingly. And there are technical reasons for that. That would be an entire talk in and of itself. And it's based off of different technologies that all have a limitation. And there are other sequencing technologies that have been able to start reading longer pieces of DNA. But in a sense, the reason why we can read short pieces of DNA but not an entire genome all at once is that you can think of the little nucleotides as having voices. And the fewer voices that you have, perhaps the easier it is to detect them. Whereas once you go into an entire choir, you really only you start to hear one voice if everyone is singing at the same time. Okay, so what researchers do, though, because they can read the short pieces of DNA but not long, is they take a sample that has multiple identical copies of a genome, and they shatter that genome into reads. So they apply some chemical, for example, or they use a sonification method. There are a variety of ways to break all these copies of the genome into pieces. And those pieces, again, are called reads. And then there's a lab component of this, a very high-tech lab component, that reads out all these reads. So that goes in and for each fragment is able to tell you what it was that fragment is. But then it's where the computation comes in is that we've got to be able to use that overlapping information in those reads. So here you see that the orange read overlaps with the blue read in all but one letter, and the blue read overlaps with the green read in all but one letter. And in practice, these are, say, 150 nucleotides long or 250 nucleotides long. But in this case, I'm showing it just as a toy example where they're shorter. But the key idea is that we're overlapping these reads in order to reconstruct a genome at the bottom and figure out what your DNA is. And now I would ask you, what does that process of shattering a genome into reads, figuring out what the reads say, and then assemble into a single contiguous genome based off of the overlap, what does that process remind you of? And I would hope that you would say, well, really, that's just the exact same thing as the newspaper problem. So fragment assembly of all these DNA fragments, all these reads, is equivalent to an overlap puzzle. Okay? Now, this is going to be made even more complicated, as hard as that newspaper problem would be to solve. Assembling DNA from all these fragments is even harder for several reasons. Well, for one, we don't actually know anything about the language of DNA. So if we knew the newspaper was written in English, we could use all these context clues that I mentioned before. But we've only just scratched the surface in understanding exactly what the language of DNA, so to speak, is. But there are other complications as well. So DNA is actually double-stranded, and it's broken over multiple chromosomes. So your genome is broken over multiple chromosomes. So you wouldn't be able to form one contiguous book, for example, and you wouldn't know if a given fragment of DNA belonged to one strand or the other, which complicates things. We also have the issue that there may be areas of the genome where fragments there weren't as many fragments. So the coverage of the underlying genome by your reads can be kind of imperfect. And then the machines themselves are going to make errors. And they're not going to tell you that they make an error, they're just going to make an error. And so we don't know, if we're given a read, whether or not we can trust it. Over time, the machines have gotten a lot better, but they still do make errors. And so we will need to know, how can we figure out if there's an error in a read if we don't know what the answer is in advance? So that makes things really tricky, and this problem is very difficult, and it's hard enough if I make some assumptions. So I want to make these assumptions and see kind of how we start taking the first steps to solve these problems. And the assumption that I would make first is, let's assume that DNA isn't double-stranded, it's just a single strand. So all of our reads came from one strand of DNA, and it's a single circular chromosome, which is a good assumption for when we're doing bacteria, which usually have a single circular chromosome. And we're also going to assume that there are no breakage points in terms of where the reads were picked up from. So we get reads from every area of this genome. So every location we're going to have a read. And let's assume that the data is good. So we're going to have error-free sequencing machines. And our aim there is we want then to reconstruct a shortest possible circular genome that's going to contain all of our reads. So here's a toy example where I've say that regardless of what lab method we use to identify reads, say our toy example has reads of length 3, and we want to reconstruct the shortest possible circular genome that contains all of them, since if we identified a read, presumably it came from somewhere in the genome, and we want to kind of get all of these reads into one genome. So I would say I'd be really impressed if I've never seen anybody who could solve this problem straight away and give me an answer of what the genome should be. So even though these reads only have length 3 and there's only 10 reads, the answer is far from obvious when you look at it. And so how in the world could we solve this problem if we had 1 billion reads? Certainly we're going to need a computer, but we're going to need to tell the computer some method that's very precise in order to solve this problem. So the question is, what method would you use? I'd have you think maybe for a second what method you would use to do this. And normally when I talk to people, an idea that they come up with is, well, why don't we just look for the maximum overlap? Okay, so let's start building a string based off of the maximum overlap. So if we start with, for example, let's start with AAT, okay? And then the question is, well, which read should we pick next? So AAT is going to need to overlap with something to its right. So we need to find something that starts with AT, so we could look for ATG. And there's only one possibility there. So we say, okay, we know AAT overlaps with ATG. So we're going to start building the genome. But then we say, okay, what's next past ATG? Well, it's going to have to be something that overlaps with ATG and starts with TG. So we're going to say it's TG something. But now we have a choice to make. So we could say, well, it could be TGC or it could be TGG. And we have to choose between those two options. So maybe we could say, well, we got lucky and we picked TGC, and maybe that's the right one. So just assume that was an isolated occurrence. But again, then we see a choice. So we pick TGC, and now we get GC something. And then we have two options, GCG or GCA. So that makes things a lot trickier. If we never had an option, then we could just reconstruct the string one read at a time, walking through and taking the overlap every time. But once we have a choice to make, then we almost have a branch in this path. And then we have, if we continue down that branch, we've seen that the path branched again. And so the issue is that branching means that we're going to have a lot of different possibilities that we have to consider. And the reason why that branching occurs for this example is that 50% of the human genome is made up of repeats. Okay, so the repeated strings are what's causing the branching, and so half of your genome is repeated strings, or that repeats that occur multiple times, say with maybe minor variations. And the reason why this makes assembling a genome that much trickier is that it's just like a jigsaw puzzle if you have repeated information. Repeated information in a jigsaw puzzle makes that puzzle really hard too. There was a popular example of puzzles from about 20 years ago that were called triazoles, and they only had 16 pieces in them. So they seemed really easy to solve, but the issue is that all the pieces had the same shape, so they were all triangular. So you couldn't use any shape information to reconstruct the puzzle. And the pieces were divided, so there was some figure, in this case they are frogs, so the frogs were divided in half. And so there were multiple different pieces that could possibly fit together. And so you might start completing the puzzle and you might get 12 of the 16 pieces in, but then the last 4 wouldn't make any sense. So you'd take all the pieces out or some of the pieces out and start trying other possibilities, and then you'd get 13 in, and then you take some out, and then you'd get 14 in, and then you take some out, and then the best you could do would be 13 again. So you wouldn't be able to get those last 2 or 3 pieces to actually fit in and make sense, and it was because you had so many different possibilities at each step. So you had different choices. And that repeated information is what makes that Eternity 2 puzzle that I mentioned earlier so difficult to solve. So I've got the Eternity 2 puzzle with me, or a smaller version of it, and I thought I'd show you exactly what I mean there. So you have the gray boundaries have to be, or the gray pieces have to be in a corner. And then so we have this piece, but the issue is that we have a pattern that's divided, it's symmetrical, and it's been divided in half. So here we have another possibility. So we could put that, for example, there's our corner, and we would need to start extending it. So we could say, well, it's not going to go like that, but it could go like this and fit together. But then the issue is, well, we have multiple different possibilities for that piece. Here's another one that's different, but that also fits in. Here's another one that is different, but that fits in, but it doesn't fit. Yeah, it fits there. And that's another one. And then we have all these other possibilities that could extend this piece down here, and so on. And there's several of those as well. So we don't know which of those is right because they're different. So we don't know which path to go down. So immediately we start to see this branching again. Okay, and that branching is what makes both of these puzzles, as well as assembling a genome, really complicated. And so we need to use a more precise, a very technical method in order to be able to figure out what it was that the genome said. Because if we just try walking down a path, we're going to get stuck. And so what we're going to need to do is come back to some mathematics and some precise computational methods in order to solve this problem.